My name is Peyton LaBonte, and today I'll be talking about my project entitled Constitutive Expression of Picrophilus Torridus Superoxide Dismutase in Tobacco. As for some background in my project, as we all know, we are experiencing rapid climate change, and plants must be able to adapt to this. They must be equipped with protection mechanisms. The organism Picrophilus Torridus is a Euryarchaea that is found in volcanic steam vents. This organism is thermoacidophilic. This means that it can survive in very high temperatures and very low acidic pH levels, and has an intracellular pH of around 4.6. So this organism contains the gene PtSOD, a superoxide dismutase gene, and this has the potential to enable plants to resist oxidative stress. Superoxide dismutase are antioxidant enzymes. These are known to destroy reactive oxygen species. Reactive oxygen species are caused by environmental stressors such as heat, and at high levels they can have negative impacts on plants. So reducing levels of ROS is important for optimal plant growth and yield. The model organism that we decided to use is tobacco, specifically Nicotiana benthamiana. This is a popular model organism used in the fields of plant virology and plant biology. And we decided to use it because of its ability to be transformed and regenerated efficiently. Our hypothesis is that introducing the pTSOD gene into tobacco will lead to increased abiotic stress tolerance. To dive into a bit of our methodology before I get into the experimental design in more detail, our pTSOD expression cassette was cloned into the modular vector pSAT1, and then it was removed from this vector and inserted into the binary vector pPZP, which was then transformed into E. coli. Confirmation of successful transformation was done via colony PCR and restriction enzyme digest. The positive clones were cultured in broth and transformed into Agrobacterium, and then via Agrobacterium-mediated transformation, they were transformed into tobacco, which was grown in the greenhouse, and once mature enough, genomic DNA extraction was conducted to screen for positive lines. The presence of pTSOD gene was confirmed in some lines. The seeds from the T0 lines are being advanced, and eventually the plants will be subject to heat treatment. Some more detail into experimental design. We started by growing our plasmids, both pSAT1 and pPZP NPT2, and we purified them via mini prep. And then we used restriction enzyme digest using the restriction enzyme ASC1 in order to remove our expression cassette from pSAT1 and open up the backbone vector and binary vector of pPZP. We wanted to confirm this digest, so we ran it on a gel and we purified the expression cassette via gel purification, and we purified the pPZP via PCR purification. Then we conducted ligation reactions. We did 3. We did one without an insert. We did one with an insert to vector ratio of 2:1, the other ratio of 4:1. Then we did transformation into E. coli. We did 4 transformation plates. We did a positive control, a negative control, and then we did ligation 2 and 3. And then we did colony PCR and gel electrophoresis in order to confirm transformation. We then cultured the positive colonies, purified them again via mini prep, and then we ran another restriction enzyme digest in order to confirm the presence of our insert. Then we made a glycerol stock of this E. coli containing our gene, which we then transformed into Agrobacterium, where we made another glycerol stock of the Agrobacterium containing our expression cassette. And then we transformed this into tobacco via Agrobacterium-mediated transformation. We then conducted genomic DNA extraction to screen for positive lines. We are currently harvesting seeds from those positive lines and will be exposing the lines to heat treatment. As for our expression cassette, we used the cauliflower mosaic virus 35S promoter. We used 2 of these, the translation enhancer, our pTSOD gene, and then the cauliflower mosaic virus 35S terminator. As for our preliminary results, we conducted genomic DNA extraction and then PCR using the specific primers of pTSOD gene in order to confirm the presence, and we did confirm its presence in a few of our lines, our T0 tobacco lines, which will eventually be advanced to the T1 progeny and subject to heat treatment. So these seeds are currently being harvested from the transgenic lines in order to germinate them, the transgenic seeds, and then our T1 and T2 lines will be characterized using various physiological and molecular techniques. So we expect to identify heat-tolerant transgenic lines. And as you can see here in this figure 1, the image displays our pTSOD T0 lines 1, 2, 4, 5, and 6. And as you can see, lines 1, 5, and 6 all are positive, showing that they contain our pTSOD gene. I want to acknowledge the biology department for receiving the Biology Undergraduate Research Award in both the fall of 2020 and spring of 2021. And that is also thanks to the Linda West Little Endowment Fund. And I wanted to thank Dr. Osena, Dr. Salihin, and my colleagues for all of their help. Thanks for listening.